Hello students, I am Dr. Arti Kumari, Assistant Professor, Department of Biotechnology, Patna Women's College. Today we will discuss another topic in immunology, which is antigen processing and presentation. After understanding the concept of antigen antibody, let us understand how an antigen is recognized by immune cells and what are the preliminary action taken by immune system to mount an immune response towards an antigen. Antigen, as we have understood, these are large macromolecules and thus can't be recognized in whole by the immune cells. The immune cells recognizes only a part of the antigen, which is also called as epitope. We have already discussed the epitopes. These epitopes are presented by MHC molecules to the T cells. Prior to put antigen to the T cell, it is processed and presented by antigen presenting cells, which is along with the MHC class 1 and MHC class 2 molecules. Experimentally, it is also proved that T cells only recognize digested peptides. There are two pathways of antigen presentation. The old cells, virus infected cells or tumor cells, these all kind of antigens are considered as endogenous antigens, and they are processed via a pathway called cytosolic pathway, and finally processed peptides are presented along with the class 1 MHC molecules. Whereas the antigens of exogenous origin are processed through endocytic pathway, where antigens are first internalized by endocytosis or phagocytosis and finally presented along with class 2 MHC molecules. Now let us understand the mechanism involved in each of these pathways. First of all, we will discuss the cytosolic pathway for endogenous antigens. The proteins to be degraded are often covalently linked to a small protein called ubiquitin. The ubiquitin tag determines the protein molecule to be destroyed. This whole process is an ATP dependent process in which a ubiquitinating enzyme complex links several ubiquitin molecule to a lysine amino group near the amino terminus of the protein. These proteins are directed to ubiquitin protein complexes, and the degradation occurs within the central channel of proteasomes, generating a variety of peptides. These peptides are further transported to the rough endoplasmic reticulum via TAP transporter proteins. The TAP designates transporter associated with antigen processing. TAP is a membrane spanning heterodimer consisting of two proteins, TAP1 and TAP2. In addition to their multiple transmembrane segments, the TAP1 and TAP2 proteins each have a domain projecting into the lumen of the RER and the ATP binding domain that projects into the cytosol. The class 1 MHC molecules are also assembled during this process inside the lumen of rough endoplasmic reticulum. The newly formed class 1 alpha chains associate with calnexin, which is a molecular chaperone in the RER membrane. The subsequent binding of the beta 2 microglobulin, which is the another chain of the MHC class 1 molecule, it releases a calnexin and allows the binding of the chaperonin named as calreticulin and tapacin, which is associated with the peptide transporter TAP. This association promotes binding of an antigenic peptide, which stabilizes the class 1 molecule peptide complex, which allows the release of the MHC1 molecule along with the peptide from the RER. Now coming to the second pathway of antigen processing, which is called as endocytic pathway. The antigen presenting cells, which is commonly called as APCs, can internalize antigen by phagocytosis or endocytosis or by both. The B cells internalize antigen very effectively by receptor mediated endocytosis using antigen specific membrane antibody as the receptors. The antigens are encircled in clathrin coated vesicles, which forms early endosomes where the pH is 6 to 6.5, where the receptors get separated and recycled towards the membrane. The proteins after endocytosis is degraded by acid dependent proteases in the endosomes, and this process takes almost an hour. The nascent MHC class II proteins in the rough endoplasmic reticulum has its peptide binding cleft blocked by the invariant chain, which is a trimer. It prevents from binding the cellular peptide or peptides from the endogenous pathway to the class II MHC molecule. The invariant chain also facilitates the MHC class IIs and exported from the endoplasmic reticulum in a vesicle. This fuses with a late endosome containing the endocytose and degraded proteins. The invariant chain is then broken down in stages, leaving only a small fragment called as clip. The full form of the clip is class II associated invariant chain peptide, which still blocks the peptide binding cleft. An MHC class II like structure, HLA-DM, removes clip and replaces it with a peptide from the endosome. The stable MHC class II is then presented on the cell surface. This is all about the antigen processing and presentation. Understanding this concept will help you all to understand the further mechanism of humoral and cell mediated immunity. So in later classes, we will read about the humoral immunity. Thank you so much.